In the beginning of my talk, I'm going to express my bias as a protein biochemist. So I'm going to hem and haw about the importance of proteins in biology, and my point will be that there's a lot of potential there to monitor disease and to come up with diagnostic biomarkers. And then I'm going to give you some examples from our work in thyroid cancer, which was the work that was funded by this grant mechanism. So the central dogma of molecular biology put forth by Crick and colleagues is that DNA makes RNA, RNA makes protein, and protein makes us. Right? So the point is, proteins are the functional elements in the living organism. The DNA has the code, the information necessary to conduct life, but the proteins are the molecules that are actually going out and fulfilling those functional roles. So they serve as catalysts. They are structural elements. They're your hair, they're your skin. They're signals, as we just heard from Dr. Graham. They're receptors. They're intracellular signaling components. So they communicate between cells. They're intracellular components, signaling components. So they also communicate within the cell. And they act to regulate gene expression, and they also modify and manipulate DNA and RNA through processes such as DNA replication, recombination, and splicing or editing. And it's interesting that most drug targets are proteins. And even as Dr. Shapiro suggests, proteins can act as drugs. Proteins and peptides can act as drugs. And most biochemical tests for disease in the clinical arena are based on tests, quantitative protein assays. So I'd like to use this slide to sort of illustrate the importance of proteins. So the genetic sequence of these two organisms, this organism doesn't change when it goes from looking like this to looking like this. So the things that take it through metamorphosis are the proteins. The protein levels change, they affect the genetic expression, and you get this dramatic change from this organism to this phenotype to this phenotype. So other examples of when we expect proteins to change are before and after disease onset. So after you get a disease, we expect there to be a certain subset of proteins in your body, of which there are about 1 million, to change as a result of that disease. As the disease progresses, we also expect levels of proteins in your body to change as a consequence of therapy. So if we treat your disease, we expect another subset of proteins to change. Due to environmental influences, if you smoke, if you're exposed to lead, if you are exposed to other toxins, and certainly during pregnancy and the menstrual cycle and metamorphosis, as I've exemplified. So all of those characteristics, all of those scenarios where proteins change lead to opportunities to find protein biomarkers that are indicative of those changes. So a biomarker is a characteristic that is objectively measured and evaluated, and it is an indicator of biological processes, both pathogenic and normal, and it can also indicate a pharmacological response to a treatment or therapy. So there are about 150 different protein biomarkers that are currently used in clinical medicine. And they can do all sorts of things, like indicate whether you've had a heart attack or how your heart is functioning. They can indicate endocrine disorders, including infertility. Diabetes is regulated by insulin. Pregnancy, the human chorionic gonadotropin is a protein, peptide, which within days of conception you can measure circulating levels, changes in circulating levels of pregnant mothers within days of conception. An event that takes 10 months, the protein responds in a matter of days. So that's a really good example of a very sensitive biomarker. And there are also biomarkers of all sorts of cancers. So in my lab, our focus is on finding new biomarkers. There are a lot of biomarkers in use, and The way that we do that is we take samples that are clinically derived, so samples from a healthy population, samples from a diseased population, samples from, in this example, benign thyroid tumors, and samples from malignant thyroid tumors. And we isolate as many of the proteins as we can. And we compare those two samples and determine if there are proteins that are indicative of malignancy, or that are indicative of benign disease, or that are indicative of healthy thyroid tissue. And we try to use those as biomarkers. So the sort of field that I would describe myself as working in is known as proteomics, and the word is actually an extension from the phrase genomics, which was the process of defining the entire genetic makeup of any biological system. So the proteome is the expressed protein complement of a cell, organ, or organism. So in essence, it is all of the proteins in your body. It is incredibly complex. It's about 1 million different proteins. So well over an order of magnitude more complicated than the genetic material. And what we strive to do is identify as many of those proteins in the samples as possible, and also quantify their relative amounts in different types of clinical scenarios. So a typical biomarker experiment or research project in our lab will look something like this, and you can do this with genetics too. So we'll start with 2 different states. That can be disease versus healthy. It can be young versus old. It can be pregnant versus non-pregnant. It can be toxin exposed versus non-toxin exposed. Any scenario that you can think of. We isolate and identify as many of the proteins from those samples as possible. Again, we compare their relative amounts in these 2 different states, and we discover a set of proteins that is either more abundant in this state or this state. And so we call those now putative biomarkers. So we've done this study by necessity in a very small number of samples, because these experiments are very time consuming and they're very expensive. So the price that we pay for being able to look at a lot of different proteins at one time is cost and throughput. As we move down this sort of translational pathway, we strive to reduce the number of biomarker candidates, right? So here we start looking at thousands. We discover tens, maybe hundreds of different proteins that are different between the 2 populations. We then use totally orthogonal analysis methods to verify these findings, and we try to move them into routine clinical assays and eventually into clinical applications where we can now, in a high throughput manner, look at individual proteins. So with a more traditional ELISA assay or a more traditional immuno approach, we can now do this in a very high throughput manner and look at lots of different samples, thousands of samples. And we can start to define population statistics. So here we're just looking at a small number of samples. Here we're looking at thousands. And right now with the Thyroid project, we're developing these assays. We've discovered a whole series of proteins that are indicative of healthy thyroid tissue, benign thyroid tissue, and malignant thyroid tissue. We're now developing those assays and trying to move them into clinical applications. So there's a diagnostic dilemma in thyroid cancer, and that dilemma is, do I have cancer or not? So there are about 300,000 patients in the United States that present in the clinic with lumps in their thyroids. Fortunately, only about 5% of those have thyroid cancer. The problem comes in when you consider the test that is used for diagnosis. The test is known as a fine needle aspiration biopsy. So they lay you on the table and they stick a needle into your thyroid, or where the tumor is, and they draw out cells and fluid. And then a pathologist looks at that and says, based on his expertise, you either have cancer or you don't. That, depending on where you are, up to 40% of the time, that pathologist will not be able to tell you whether you have cancer or whether you'll be fine. So there's the diagnostic dilemma right there. And the standard practice is, if you're one of those people that the pathologist can't tell you, you have your thyroid either partially or fully removed as a measure of caution. So they don't want to miss cancer, right? Turns out 80% of those indeterminate cases are benign. So in 80% of the cases, they removed your thyroid unnecessarily. So there is definitely a current clinical diagnostic problem. It's the procedure is invasive, it's costly, and in many cases it's unnecessary. So our project aims at that right there. Our project aims to increase the diagnostic sensitivity and specificity in thyroid cancer and eliminate as many of those unnecessary surgeries as possible. So we've done some studies where we have defined the proteins that are unique to benign and malignant tumor tissues, or that are overabundant or underabundant in those two types of tissues. We've published those results, and we predict that, and we're in the process of, and that's what the Tech Transfer Office in the state of Colorado funded us to do, is to now go and test with more routine clinical assays, ELISAs, whether or not they can offer more diagnostic power than the current test, which is the fine needle aspiration biopsy. So the potential for impact from our side is, I think in the worst case scenario, about half the time you can't tell whether it's cancer or whether you'll be fine. So I think we have some pretty good chances to make an impact here. I suppose in the worst case scenario, our protein biomarkers could be sort of a complementary strategy. So only in the indeterminate cases, we could make measurements in that tissue and fluid that's drawn from the thyroid on the certain biomarkers that we've proposed, and make an indeterminate diagnosis and determined diagnosis. If our markers turn out to be more accurate, faster, or cheaper than the fine needle aspiration biopsy, then our tests could actually replace the need for that pathological and subjective diagnostic criteria. And the last one is sort of optimistic, but not entirely outside of the realm of possibility. So if those markers that exist in the tumor end up getting into the circulation, the general circulation, and we can do a simple blood draw instead of having to take samples from the actual tissue, then we could talk about doing a screening sort of test, where instead of just having people present in the clinic, we could actually develop a screening test for thyroid cancer. Just to sort of continue on with the potential impact, if you just consider that, if you take about 40% of those, about 60,000 surgeries are performed in the U.S. Every year that are totally unnecessary, and that's a couple $100 million worth of unnecessary surgeries. The cost of the FNAB and the pathology is about $400. The cost of what we propose to do would be anywhere from $5 to a couple of $100, depending on how many markers we find that are advantageous. But it's important to say that we don't know which of these, or if any of these, are going to be any good. That's just the potential. And we are in the process of determining whether or not this is going to be a reality. We've done this in tissue. We are currently acquiring the fine needle aspirates to test our biomarkers in those, and we're going to also acquire the blood samples and test our markers in blood. So I certainly didn't do all this myself. I'm not a clinical endocrinologist. So Dr. Haugen is the head of endocrinology here at the university, and he is one of our collaborators. Mark Duncan is a colleague of mine. Dr. Natia Ermer was a clinical fellow from the Netherlands, and of course the funding source, and you guys. And I'd be happy to take any questions.